Welcome, everybody, to the session number 10 of AI Solution for Sepsis at the 5th World Sepsis Congress. Good morning, good afternoon. I'm Katherine Urbáez. I'm the Executive Director of the Health Diplomacy Alliance, and we are thrilled to have you here for what is going to be a promising and dynamic and forward-looking discussion on this session. As you know, sepsis is a global health crisis, claiming millions of lives each year. Those numbers not only underscore the urgency, but the early need for early detection and timely intervention, where AI plays a pivotal role in transforming sepsis care. For this discussion, we encourage you to use the chat and actively participate in the discussions. For any question, please submit your questions in the chat using the prefix Q, and we will integrate these questions into our discussions with the speakers as we move forward with them. Today, we will explore several different aspects on how artificial intelligence is transforming sepsis care, from predictive algorithms in emergency departments to AI-assisted diagnostic in intensive care units. Our speakers will showcase real-world application, the challenges in implementation, and the potential of AI to reduce mortality and to improve clinical outcomes. And for that, I want to open the floor and the discussion with Dr. Nathan Shapiro, who is professor of emergency medicine at Harvard Medical School and an attending physician at Beth Israel Deaconess Medical Center. His research interest is mostly focused on diagnostic and treatment of early sepsis and resuscitations in critical illness. And he's going to be covering today about the Sepsis ImmunoScore, the first FDA-approved AI tool for sepsis prediction. Dr. Shapiro, you have the floor.
Thank you very much. As mentioned, I'll be talking about the first FDA-approved AI-based score, just in the way of disclosures. Prognosis is the company created the score, and I am a consultant for them, just for full transparency. So let's just think about big picture in general. As a clinician, when we go and see a patient, we're going to then integrate what's going on with the patient. We're going to think about the demographics. Perhaps we're going to get some biomarker measurements, standard lab testing, vital signs, comorbidities, and I'm going to add, not pictured here, physical exam. So these things combine to help us assess the patient. And I think we're asking, in some ways, I like to break it down to two basic questions. Number one is diagnosis. Is it an infection or sepsis that's making the patient in front of me sick? And number two is prognosis. Okay, I've decided that they have an infection maybe. Are they high severity or low severity? And what kind of care do they need beyond anti-infectives in order to support them? Is it care that they can just go home and stay well hydrated? Or do they need to come in for intravenous fluids, perhaps respiratory care, etc.? So the Sepsis ImmunoScore, which is going to be the topic of the conversation, is an AI tool. And what this AI tool does is it taps into the EMR, electronic medical record. It pulls comorbidities, standard lab testing, vital sign, demographics, and comorbidity results. It pulls them in, and then we add in a few extra protein biomarkers. And the ImmunoScore is an AI tool that integrates all of these things in order to predict whether a patient has sepsis, and then if they have sepsis, is it likely to be low or high severity? So the way that we submit the Sepsis ImmunoScore will help is it's an aid to the clinician. So the clinician sees the patient, makes their judgment. I like to call that human intelligence, integrating all of these things, and then adds to it artificial intelligence, the Sepsis ImmunoScore, and the combined decision-making between the clinician plus the knowledge of the Sepsis ImmunoScore should help for an improved assessment of sepsis versus non-sepsis and low severity versus high severity. So let's drill down on this a little further. The proposed workflow: patient enters the hospital. The clinician says, I have enough of a suspicion for an infection that I'd like to go ahead and get the Sepsis ImmunoScore to help me. The EMR data is streamed, proteins are assayed, and then it gives the output is the likelihood of sepsis, very high, high, medium, and low, and that also corresponds to the severity. We published this in the New England Journal of Medicine AI in 2024, and I'd like to go through the manuscript as I think this is the best objective demonstration as to what we did. So the objective was to evaluate the performance of the sepsis ImmunoScore. We had two endpoints. Primary endpoint was sepsis defined by sepsis 3 within 24 hours. The secondary endpoints were length of stay, ICU admission, mechanical ventilation, and vasopressor use. Prospective observational study. And the design of the study included three independent cohorts. The first one was the development cohort of 2,300 patients that was used to create the AI-based score. There was an internal validation just to make sure the score was working well. And finally, the score was locked, and proof of concept was the external validation of 700 patients. And these came from 5 different hospitals. So our derivation cohort is here, 2,890 assessed, 2,366 enrolled, and then you can see the internal validation and external validation. Just to give you a feel of the kinds of patients that were being enrolled, they're enrolled across these, it was 6 different hospitals, mean age of about 64, kind of common things you'll see in sepsis in the United States, which includes a high comorbid burden of CHF and diabetes at about a quarter of the patients. So these are the components that go into the score, kind of things we typically associate with sepsis, but these are the different types of inputs into the score, including vital signs, CBC parameters such as platelets, BMP, and then finally procalcitonin, C-reactive protein, and lactate. So the methodology is we took all the EMR data available, narrowed it down to these are the key components that are going to drive the score, create the sepsis ImmunoScore algorithm that then gives the output of risk. This is a SHAP value demonstration, so this is where we get into some of the machine learning fancy tools, but the idea is that this shows the things across each patient that tend to drive the risk. So the top driver is actually procalcitonin, but what I also found interesting, which correlates with studies that have been going on for decades, is respiratory rate, blood pressure, and platelets are actually other key components driving the score, which when you look back at studies over the decades, respiratory rate and platelets especially are typically key drivers for sepsis. So there's some sort of proof of concept validity. So here's what it looks like. When you go into the low, medium, high, and very high categories, this was the derivation set, internal validation set, and external validation set. So you can see a nice step up of risk for sepsis based on the score. The AUC is about 0.8 across the board. So just looking a little bit more at the external validation, if we look at likelihood ratios, so, something 0.1 is deemed as excellent for negative predictive, and then once you get to the 5 to 10 range for positive predictive, also found to be of good value. And if you just look empirically, it's 3% risk of sepsis versus 13%, 37%, and then 69%. So again, we were happy with the risk stratification that occurred. And then when you look at critical illness and mortality based on these risk groups, you really see again a nice stratification of risk for these types of comorbids. So we would submit that these categories are helping to demonstrate low risk versus high risk for severity. So just to kind of pull it all together, it'll flow with a holistic data input. We take the different categories of inputs, CRP, procalcitonin, lactate, vital signs, existing labs, demographics, and use that to synthesize the score. Just to demonstrate a little further, one of the criticisms of AI tools is the black box phenomenon, which is the inputs go in, the output comes out, but it's not really transparent to the user. So in order to overcome that limitation, we use these SHAP values, which for an individual patient, it says, here's what the person had, and this is what's driving this risk score. So the overall score for this one patient is a high risk, 86. And then it's saying, let's be transparent on a high lactated platelets that are pushing this a little bit. In this particular case, it's not as relevant because it's such a high score. But for a low risk patient, you would see these green things coming way over and saying these are the types of things that bring the score down. So the idea is this patient scores in the high risk category. Here's the components and to the clinician so they understand why it's high risk and then can act accordingly. So that's my brief tale of the first ever sepsis ImmunoScore. There's a licensing agreement with Roche, and the idea is that the score can be integrated into the EMR to make this available to assist clinicians in diagnosing and prognosing sepsis. And with that, I thank everyone for their time, and I'm happy to take any questions.
Thank you very much. And that's really an interesting perspective in terms of the AI tool. I have myself a questions as participants, they cheer up in the chat. I would like to know, I mean, you have mentioned about the blackout and how does that tool integrate with other existing health records in the health systems. But how do you see that this can provide real-time decisions in terms of if the data has been collected appropriately or not the data that that the AI is collecting, but if the data has been correctly put in the system by the clinicians before the AI comes into place?
So it's a great question, which is, do we have all the data? So the way the tool works is there's required elements and non-required elements. So there's a couple key required elements. If it has all the required elements at the point that it feels like it's prognostic, it can make its decision, it'll make it. If it's missing the elements, it will alert the clinician to say, listen, we're missing a CBC. Please order a CBC so that can get added, or we're missing a procalcitonin. Please order this so we can give you the sepsis ImmunoScore. But very good question. Thank you.
Excellent. And I assume this can be used both for adults and for children, as we know that there is quite risk. Yeah.
Yeah. So this particular score was only derived and validated for adults. So it's definitely, the future, I hope, will hold a pediatric ImmunoScore. But for now, I think it would be fair to say you want to only use a tool in the population in which it was validated. And so for this population, it is, in fact, adults.
Excellent. That really does make sense. I think on this subject, we're going to continue on questions that are going to come up in the chat, and I myself have more questions. But in order to follow the next speaker, Dr. Sujoy, who is the Chief Medical Information Officer and the Vice President of the Apollo Hospital in India. His current research and ongoing research interests are in developing and adopting artificial intelligence in healthcare and digital platform. And he's going to be speaking with us about using AI for early detection and predictive modeling of sepsis. So Dr. Carr, you have the floor.
Thank you, Katherine. It was truly a pleasure to hear from Dr. Shapiro about the work that they're doing on ImmunoScore. And I'll just go about it, sharing some of the work that we have been doing at Apollo hospitals to actually, bring in an AI-based systemic approach to early sepsis detection, recommendation systems, and blood sepsis study, which is a consortium work that we've been doing. And of course, we'll talk about our journey for accreditation. And at the moment, we're going through multiple peer reviews for the database that we have, both from Apollo and then our external validation microbiologist, and also have practiced infectious diseases at hospitals. But for past 7 side, with the data at Apollo hospitals and building consortia to actually work through. So if you look at, from a sepsis perspective, it's a, systemic disease, which is, not a single disease condition. And for us, the hypothesis and exploration to actually look at it from an opportunity to map the baseline phenotypes and risk for progression to sepsis, including stratified to sex, age, comorbidities, clinical stage, frailty, and others. And that's exactly what we're doing in adult EWS triphasic solutions, where we have multiple things that we identify from an early deterioration. But sepsis is one of those key, markers that we have looked at over a period of time to bring in to accurately initiate antibiotic to not only diagnose sepsis appropriately, but also put it in, how you go about initiating an antibiotic therapy, which possibly goes about in days or next 10 days, about the outcomes that you are associated with therapy associated with infections. And lastly, we're going to talk a little bit about the work that we have been Cambridge and the rest of the other people about the baseline full blood count morphology and response to challenge that will differ in different phenotypes, and that would call it very similar to what you just heard. And this is about in that we took about close to 145,000 patients from 2018 to 2023 with 32 million data points, looked at the vitals, demographics, comorbidities, diagnosis, their post-surgical perspective, and discuss about these 4 categories, and look at it out from an output from a prediction of sepsis, sepsis-related mortality, risk stratification for clinical deterioration in sepsis, and identify in the very same way that you saw in terms of SHAP values for both XGBoost and then feature importance for the work that we do on the deep learning side, advice for monitoring into clinical decision support, and then go about and level up or down protocols for doing that. And we've been doing this in a standalone app, telehealth EMR, that we have been doing because we have had this work under patent. So, it is not publicly available at this point in time, though it's going through peer review and validation at multiple centers, and we'll come back with the results very soon. But as you can see it from, different works that we have been doing on clinical features and survivability analysis for, sepsis-associated renal failure or pneumonia and sepsis cohort, and then studying the variations of each one of these diseases and how do they go about in looking at. So we looked at about close to 102,000 people over a period of time, starting from about close to 10 hospitals in Apollo ecosystem. Apollo Hospitals is one of the largest hospitals group in India, and so it took about last 10 years of data to actually 102,000 people with 25 plus features, very akin to what you were seeing in ImmunoScores, with a validation set of around 43,000 with 25% features with alerts for clinical deterioration and for sepsis and risk of mortality in 7 days, with an event rate of close to about 3% and about positive predictive value close to around 40%. And as you can see from this, that our AUCs were slightly higher because of the volume and the sensitivity, the specificity we were getting at. The sensitivity still is around 0.85. The precision recall comes around close to about 0.74 to 0.7778. And we had to do about going around the threshold curation to see whether you were screening more people to have more sepsis detection, or you were looking at it much more pinpointed diagnosis to go about in this. What's interesting is that we also went about in doing the scoring around Matthew correlation coefficients to understand whether it is a clinical grade product or not. And we find it to be close to around 0.7, which actually closes to where we are in terms of, putting it into the clinics and doing that. And this is exactly what you are seeing in a kind of a dashboard. So this is a prototype dashboard, but the actual dashboard looks pretty better than this, where you have all the vitals of these patients, just like what you saw in the previous presentation as well. Coming in through that, you would have the in a way where you would look at these vitals that have been streaming from patients. So we have the ability to look at about 1 million data points coming in on a daily basis to actually process that, and then go about and building a 24-hour circles to actually see how many of these patients would have sepsis-related mortality or risk of mortality or risk of sepsis going about in next 24 hours. And then if you click on them, then it would go about in providing you with the SHAP plot, which would actually go about in giving you what are the factors that are bringing it down, what are the factors that are pulling that patients up, and how do you go about in looking at it from a deterioration perspective. And as you see it from the vitals, all the 6 vital signs, and also look at it from a clinical deterioration based on 4 hours, 4 hours, 8 hours, 12 hours, or 12 hours, and look at it from every hourly fashion to see whether it is progressing or whether, if the patient is progressing or they're deteriorating over a period of time in doing that. And you look at it and, take your judgment from a clinical perspective and take it from there. And then you have also in the same dashboard, you would have the lab markers that are ingested in the algorithm. Sometimes you may not have labs come in about in every little time that you want them. So possibly in labs would only get in once in 48 hours or others. So if you're calculating in 24 hours, so whether it is a complete blood count or a basic biochemistry or the the coagulation profile or the inflammatory markers that are there. So you'd go about it in looking at techniques like, nested bird and forward filling to actually bring it together to break it down. And we have also gone about in looking at, the pneumonia angle to it and see how you also look at the serial chest x-rays over time. We gave it to our nurses and they find it to be a little complicated, to be honest, and then to look at it from a sepsis perspective. And then we build a generative AI on the top of it to say, you can then interact with the dashboard by putting in, vitals, labs, simulate it, and then tell what would be the possible next step. So this is the one where we are helping in detecting of the early warning signs associated with sepsis at this point of time. But going forward, we looked at our, the entire sepsis cohort, and very likely what you saw in the previous presentation, again, the age that is picked up in our study as well is somewhere near about, 65 to 74 years. Looking at both from a perspective of both the genders in that perspective. And what we have been doing is then take this, the demographic details of the patients, including the age, gender, location, whether they are at the outpatient or ward or the ICU specialties, or even, the kind of consultants who are there and treating these patients, and look at the patient profile from site of infection, what samples you are taking, what empirical antibiotics that have been given, what is the underlying condition, or what are the concomitant medications, and then go about in building a recommendation system, much into what you see in Netflix, and then go about in, providing at the point of care what would be the right set of bacteria that you are looking at. There is a separate study which actually goes about in looking at bacteria versus viral, and I'll come back to that in a moment. But this is about if you have a bacterial infection, then what is what is the possible bacteria that you are looking at in an outpatient, inpatient, or in a ward or an ER setting, and what possible antibiotics that you're going to do. So we had to do it in a filtering mechanism, and then with the secondary data, we had machine learning on top of it, use cosine similarity to identify what would be the samples that are coming from each one of these specimens, and then go out and looking at it, this cosine similarity for each one of these samples, plus the sites they are coming in and what type of ward, ICU, or the outpatient setting that we are looking at in order to build this. So we looked at about close to 360,000 isolates. So one of the things in India is that you could look at around a lot of these data with about 7 to 8 input variables, 57 specimens, 180 organisms, and possible combination of 141 antibiotics at this point of time. I got an AOC, ROC for about 0.866 with a pretty good, decent precision recall to actually go about in doing that. And then what it does is then go about in looking at any different combination of antibiotic and organism, and then say how many of them would be resistant to the what would the sensitivity in the wards, or what would be the sensitivity in the ICU to go about in these patients. So what it actually helped us in building is this solution, what we call as the ears, where a patient with a demographic data or comorbidities and current infection would come to outpatient visit, or even in the ward when you see that, and you put all of these together and send the samples. At the same time, you can run at the back end the ER's algorithm with the descriptive statistics, the machine learning, and the recommendation systems, which will give you the top 3 organisms that are probable for that patient at that point of time. And then correspondingly, it will give you 3 antibiotics. And then you look at it also certain other factors like, what is the renal status of the patient, or hepatic status of this patient, or are we dealing with somebody who is lactating or is pregnant, and take all of those guardrails, including, and suggest what would be the best recommendation. So we did the prospective validation of this by using it in clinics and to say, what these people are doing. We're getting accuracy of around 87% at this point of time to see what is the kind of recommendation they're doing and what is the 5 days outcome of these patients over a period of time. So, and that to this, we are adding the new research that we're doing on the full blood count. And in that full blood count, what we're looking at, and and this is a work that we have been doing with our consortium at, University of Cambridge to actually look at the layers of complexity. And this is very well published now. And so what we have here is that, we would possibly look at the FBCs and see close to about 12 to 24 data points, depending on where you are looking at this report. But if you go about and look at the reach data that comes out from the FBCs from these machines, whether it's Sysmex or Siemens, Advia, or others, you could go up to more than 3 million data points that comes in, which would where your RBCs, WBCs, it functions, and others would actually be there around for both the entire blood count and also specific from the white blood cells. And we are actually see the imprints on these plots about for influenza, COVID-19, or sepsis, or even some other conditions related to hematological conditions or other clinical conditions like cardiovascular disease or even pregnancy or iron deficiency anemia. But what it does is specifically for the sepsis is to then identify early points where we're seeing that patients those signals are there, and we would be able to determine those signals to put it into perspective and see how do we go about in putting this into practice and bringing them about studying further. I'll end with the last slide. So we are now, medical device single audit program certified, process certified for different, settings worldwide, including Australia, Brazil, Canada, Japan, and USA for at least the first two products, where we're doing some extensive validation and peer review and coming up with the specific threshold curation and results for those particular community. I'll stop there. Thank you very much for the opportunity.
Thank you very much, Dr. Kaur. That's really interesting. And I think I'm going to go to the questions, but I have some curiosity on understanding the efficiency of having the full blood count in terms of this complexity on detection. But I will build up on that. If we go to the chat, we have seen a few questions. So I think those questions are both intended for Dr. Kaur and as well for Dr. Shapiro, but I will start with the first one to Dr. Kaur. We see that sometimes erroneous data is logged and makes a sepsis alert. What is the best thing to do in terms? I don't know if this could be integrated into the modeling of predictive modeling of sepsis. What are your thoughts, Dr. Kaur?
Yeah, so of course, the kind of data, erroneous data logged into this is always a challenge that we have seen. So in our system, we have 2 or 3 things where you look at the data that is often fetched from the EMR directly to actually do that. So that's number one. Number 2 is also to get the data directly from the device. And then, you would have to have your own ways of algorithm to actually sieve through these data to understand what to pick up over a period of time, particularly, when you are looking at a constant data stream at a high velocity that are coming in and what that you would be looking at consistently as a value over a period of time. The other thing is very important around the lab, which I spoke about. So you will not get the lab values to come in every, possibly every day. And in, low resource setting, you may not even have them every alternate day as well. So one of the things is that how do you use machine learning techniques to forward field this, but of course give an alert that you require a recent value to actually calculate these early warning signs appropriately. So what we have done is in our simulation studies, we have gone about and looked at if we drop one of these important factors, what is the hazard to overall model and how the model performs in those settings. And we're trying to make it more robust by doing that.
Excellent, thank you. I think the next question is intended for Dr. Shapiro. Is there a way to use this feature? I think he's referring to the ImmunoScore for future studies in research, or is not possible due to that it's been the consent to rush?
Could you say that not possible to use in
terms of research for further
Oh,
studies?
sure. No, it's definitely possible. Roche is is distributing it as part of one of their platforms, but there's also some independence and also looking to refine the score either for sepsis or maybe even other indications over time.
Thank you. There is another one, and I think it has been touched upon, but it's how PCT is relatively expensive in low, middle income countries, but how that can affect those scores in terms of determining. I think it is for both of you in terms of determining the scores of the early detection of sepsis. I will give it to Dr. Shapiro first, and then I will pass to Dr. Carr.
Sure. If I understand it, the question is about procalcitonin and the expense.
Yeah.
I would say procalcitonin is one biomarker for sepsis. There's different biomarker for sepsis, and I think the goal is to use the biomarkers and vitals and other things you have available, and then figure out the best ways to use those things combined to complement the clinician in order to treat patients. So I think one has to use what's available. If they don't have procalcitonin, use a similar biomarker and figure out how to best integrate those results.
And the other point that I want to make it very quickly is this, that many of these biomarkers are determined by their individual hazard ratios that you would bring in for that particular biomarker in the study. So what is their hazard ratio over a period of time in terms of getting into a sepsis diagnosis, and what is their value from a diagnostic element, what is their value from a prognostic element, when you are studying that, and what part of the day, what part of the whole journey around the sepsis, where do you bring in those biomarkers, is again very, very interesting. And what we're doing with the basic count is to actually correlate with the imprints that we are getting from the signals for the full blood cells, to also to correlate it with conventional biomarkers to say, would you then go about in testing them at the right moment? And a huge database actually helps out in that.
Yes, that builds up a little bit on my question about using full blood and the cost of it. When do you see the value of going to a full blood count in terms of, is it when you predict that there is going to be a high risk, or do you follow any patterns? I know you presented it in your presentation, but I think it's interesting to understand at what moment the decision is making.
Yes, Katherine, that's a very interesting question. And my answer to this is that, we would do the CBC for any patient who are coming in, but one of the initial tests that you do is that way. So the idea is then you go about in extracting the, those signals. There are special methods to do it from the hematological machines that we have. But the thing is, all of these machines that we use, conventional machines, mostly in our labs, would be able to give you or throw you those data points to actually bring it in and do that. So you can do this again to identify early biomarkers or early imprints. You can do this also from a prognostic point of view. And we don't know yet if there are those imprints that would also go about in talking about chances of mortality or very high risk, which Dr. Shapiro was referring to.
Interesting. I think there's going to be a lot of more questions, but I will give now the floor to Dr. Ian Hosein, who is a UK and Ireland-based consultant, medical microbiologist, and visiting professor of medical microbiology at the University of West Indies, who has been working on advancing precision medicine through AI-driven diagnostic tools and target sepsis therapies. And he's an enthusiastic clinician and teaches healthcare staff that technology must be rooted in sound bedside skills and observation in clinical reasoning to achieve optimal infection diagnostic and management. So Dr. Ian Hosein will give us an overview on AI in antimicrobial stewardship. Dr. Hosein, you have the floor.
Thank you, Chair. Many thanks for this opportunity to present. And I'm speaking to you currently from Tullamore Hospital in Ireland. Antimicrobial stewardship is a vital approach to improving clinical outcomes and to reducing antibiotic pressure as a means of reducing antimicrobial resistance, which is recognized by all authorities across the world as a priority. For purposes of this talk, I'm using the word infection as the underlying problem and sepsis as sepsis syndrome. I'm also going to use artificial intelligence as a global term for the various models and processes we have, whether it be machine learning or deep learning. Where we look at antimicrobial stewardship, certainly one of the challenges that we face every day is what was just mentioned in the discussion, decision-making, the point of decision-making, when a particular decision is made to use an anti-infective agent. And one of the things I want to talk about, which has been mentioned earlier, is the value of AI. One of the purported values is the ability to handle large volumes of data, to analyze it, and so forth. So we're going to talk about data in terms of stewardship, where AI can fit perhaps with data management and where that benefits stewardship. There'll be a brief review of the literature and then some conclusions. So let's talk about antimicrobial stewardship and what we mean by that. It is certainly an approach to improve prescribing and to reduce AMR together with infection prevention control efforts and so forth. When we look at how it is structured, it reflects the hierarchy of a hospital, and my focus is on hospitals today. And at board level, we know that data is required to review financial management systems, patient safety. There may be requests to a hospital board, to what extent are you complying with national programs to reduce antimicrobial resistance, AMR? So a board needs data. At a clinical directorate or divisional level, teams and so forth, there will be audits done about prescribing practice and perhaps reports about expenditure. And data, again, is important. And certainly at team level, one of the things we notice pretty much every audit shows us is variation. Variation between teams, variation within individual attendings or consultants in a given team. So data can help us. Data without AI can help us perhaps to reduce that variation, particularly if it was integrated. So we had benchmarking data from other hospitals. We had guidelines based on data which are international or local, etc. But the heart of all of these, whether it be at the board level or team level, is bedside prescribing. And I'm going to use the word bedside prescribing to mean literally at the bedside or virtually, where we have the patient in mind, whether it be in a room or elsewhere. We have the patient in mind and we wish to make a decision about prescribing. And that really is the heart of the issue. And that happens every day. It's a very challenging issue across the entire world. And what the literature has shown over the years regarding the gap in prescribing that is valid and invalid, the 30% gap, why does that actually exist? Clinicians are trying to do a good job. People want to help their patients. So why then do they overprescribe? Why then do they use the wrong drug, the wrong dose, the wrong duration, and so forth? So certainly insufficiency of data. A junior doctor who is hard pressed is working very hard today. They don't have the radiology to their hand. They don't have the ProCal level back yet. They're making a decision. So insufficiency of data, the interpretation gap, where I think Dr. Shapiro mentioned about integrating the various strands from the health records and so on. So you find the interpretation gap between looking at one parameter, a white cell count, a CRP, a ProCal, without a holistic approach. If we had the radiology image, if we had an ID or microbiologist next to you, a junior doctor is more likely to make a better informed decision, but we don't have that. There are certain paradigms and habits which happen, and people are risk averse. And certainly from the sepsis standpoint, we do not want them to be overconfident. We want a certain risk aversion to make sure, and from the sepsis standpoint, we intervene. But all of these issues, data insufficiency, lead to overdiagnosis, broad spectrum choices, and so forth. Where can AI help? So we recognize we need data at whatever level we want. And the purported benefits of AI in terms of large volumes of data, I think as the chair mentioned, what is the validity of the source data? Even if we're exploring an electronic health record, there's variation in that record. AI is going to be very fast. It is fast now. But is clinical introspection a waste of time? The emphasis of a board is the patient flow, patient throughput. We got people coming to ED. We have people waiting for discharge. The pressure on clinicians for discharge is intense. But there is a certain gem in standing at the bedside and having a discussion and having a debate and having a disagreement. surely that is not a waste of time, but we all know it can lead to insight into that patient. There's a human tone when you read AI-driven information. It's quite endearing, but of course, we also know that AI hallucination, the term used in literature where information is presented by a system in a very confident manner, but it is wrong. But will a non-specialist know that the information given to influence stewardship is wrong? The models are meant to be trainable. Who is doing the training? And the models will give a most probable answer. Who carries the accountability for that answer? Looking at the literature, it is certainly suggesting that clinicians can be guided in their choice. And certainly there's data, I think this is fairly solid data, looking at laboratory uses where we can now predict resistance faster using AI models. And a study by Amin from Antibiotics 2023, where they compared 5 other studies, observational studies. And they do say that AI seems to be capable of improving prescribing, but they note that prescribers, the front lines, were not involved in model development, and they note that implementation also did not involve the clinical practice in real time. This is a paper by Sun et al. In Humanities and Social Sciences, and AI hallucination again is mentioned here. And they do make a plea that we will need to validate information. They say that if that information is given out, clinicians will remember that it was wrong and so affect their future use of AI. And we have a paper by Mara in Infection Control and Hospital Epidemiology. I particularly like this as a paper looking at AI in healthcare epidemiology, infection prevention control, and antimicrobial stewardship. And they talk about, again, the need to validate information. And I want to quote one of the lines from that paper, which I particularly like, which shows my own bias. A human touch remains essential to uphold the passion and humanity of medicine and should not be taken for granted. In conclusion, I've gone very quickly, but I hope there'll be some questions. AI is already with us in human society. It's on the roads. AI is in healthcare. There will be efforts to bring AI further into antimicrobial stewardship for the reasons I gave. Sepsis was already discussed. The models may help with data analysis for decision-making, the moments of truth, those critical points where we're going to say meropenem, aminoglycoside. Those are the points we want to influence. And AI certainly seems to be able to bring that data to the integrated data. However, clinical thinking, as a colleague mentioned, is extremely complex, and it is also subjective. We still look at the eyes of the patient. We still look at whether they're smiling or not to help us decide, are they improving, despite the CRP remaining static, despite the procal not being as clear as we want it to be. AI can generate information. We see that information data, I'm using it synonymously because the literature uses both terms synonymously when you look at AI data. Opportunities and challenges are there, including hallucinations. I think the final plea would be frontline clinicians regarding at all levels, junior doctors, pharmacists, must all be involved, not just the specialists in AI model development and implementation, if we want to influence behavior at the bedside. Thank you very much.
Thank you very much. That's really insightful from that perspective, from a stewardship, antimicrobial stewardship. I cannot stop and ask this question. So based on what you have said, I think it is a mix of understanding the importance of having a AI-driven diagnostic tool, but also how we compare it with traditional methods in detecting sepsis. And I think between these two things, having the AI-driven diagnostic tool and the traditional methods that you already mentioned, becomes the AI hallucination kind of phase that we have to have in consideration between both of these spectrum. So how do you see we can balance between the three, the diagnostic tool as an advancement, the AI hallucination for accurate data, and maintaining these traditional methods in detecting sepsis?
So I think Dr. Shapiro mentioned in his presentation that we still need a human dimension, human intelligence with all its complexity, where an AI or artificial intelligence could be an add-on to that. So that's one way. We need to not give up on traditional thinking. And if you recall, I used the word infection as the underlying phenomenon in sepsis as sepsis syndrome, because, sepsis, our thinking should be different in sepsis syndrome. The speed, the decision-making should be different because of the potential mortality. But there are many more patients across all hospital wards who have subtle infections, a bit of chest, bit of urine, bit of abdomen. But treating those patients can be the germinal issue in stewardship, because that leads then to more antibiotic pressure. If we make the incorrect choice, the infection may progress and then you end up with sepsis. So to answer your question, I think it was answered earlier. We need both human intelligence, which we must not give up on. The subjectivity that we have is not something that we should hold in a lower level. It is a fundamental level in decision-making when you speak to a patient, how they respond, their tone, all of that is information that we can use, which an AI system, to my recollection right now, cannot do. Thank you very much again.
Thank you. Thank you. That's really, really interesting. I'm going to go to the chat now, and we see a question is not addressed for any of the specific speakers, but it's more on the confidentiality of patients. From your perspective, and I will open for the panelists, is there any risk of data leakage when using AI tool? So it's a lot about to do privacy. I don't know who wants to take the call. Dr. Hosein, you would like to, and then Dr. Carr.
So privacy is already embedded in medicine. Although I put a lot of emphasis, as I said, at the bedside, we're always aware of what we say, what we do, how we talk, who's listening. In terms of AI, if it is brought to the front lines and we're now checking information and discussing it, I think we already deal with it. We're at a ward round where you have a computer-aided ward rounds. We're already aware. So I don't see a problem at the front line. In terms of the security, the cybersecurity element, I think that's an area that is outside my expertise. It's a huge area. Others may wish to comment, but that really is, it's a cybersecurity issue that can apply right now, even without AI.
Dr. Carr, you want to react to that?
I think this is one of the fundamental questions that we have when we are either designing, developing, or integrating or deploying AI in clinical setting. And I think it's a very pertinent question to answer, as we see there are lots of regulatory changes and bringing in the new laws across the world, particularly keeping in pace with the the technology that develops. So there are various methods at this point of time looking at, of course, over the years, the whole algorithms that are there for anonymization protocols for both tabular and textual data, and have evolved, and they're pretty accurate in the way that they are looking at, whether you are looking at it from Microsoft, Google, or John Snow Labs, or some work that we have been doing at our labs as well to do this. But I think the where it is shaping, when you are looking at quite a sizable amount of data set, where particularly we're building language models on a clinical data set or using retrieval augmented generations to actually comment from from the data sets that are already there in EMR and others. We need to build what we call as systems like confidential computing, where you have it under a canonical system. And that's been one of my active research with Microsoft. I mean, if you allow, I can just put one slide to actually describe how it works, which is about, going about in, so this is a very latest research that we had presented to in one of these Microsoft conferences last year, and then there is a full volume of paper that are coming in. So what we do is a partner tenant where they have that there is a processing of the data encrypted in such a way, and put that into a canonical systems where you have remote attestation, storage, and identity of the data, and then you would have in a layer which is ready for any API application programming interface that comes from AI from that perspective. And anyone who builds an AI tool then can go and expose this over these, or once you have the remote attestation and decryption key, to actually access this data. And those people will not be able to see the data in the structures that we normally see, but they will be able to run their algorithms on top of it, get the results, including building the models or training the models or fine-tuning the models, and then go about in getting their transaction ID or about, the parameters that you would be, those key quality indicators that are the key machine learning indicators that you would get it from a fine-tuned model. And we tested this out in EMR data, particularly also looking at data from patients, from labs, from medications that you use, and it works perfectly fine as if you are doing this in your own CPU or high-end computing devices. But when it comes to images, we're just exploring at this point of time. It works very well for X-rays and ultrasound or even for mammograms. But when we go into higher modalities, that's where we will still have to test it further to get there. I have no clue at the moment how it will behave in genomic data or videos, or in histopathology slides, like where you use pyramidal teeth and others. But that's one research that we are continuing to do to see how we can protect and do this work. The conventional way people have actually done this is by using something what we call as a federated learning models. And even the confidential computing is actually a couple of steps ahead of that.
Thank you. Thank you very much. There is another question that relates a little bit of one of the first questions in terms of the data that sometimes the collected data sometimes contains error, but how we ensure that the model accuracy is effective, and how or what are the solutions that exist for settings that do not use collection of electronic data is a question from the chat. So I think there we go back to the human aspect of it. What solutions exist for settings that do not use these electronic data collections at the healthcare facilities?
So I think we're making decisions right now. Many hospitals do not have electronically available health records. We do not lose the strengths we already have. If, again, colleagues mentioned it, you use what you have and you chase up what you don't have is what happens every day in a hospital ward. So if you don't have an EHR, we still have health records, which are paper, and they can be accessed. So they're not mutually exclusive. Medicine still goes on. We without AI, we're doing a good job across the whole world. It's a question of how we bring in this technology, I think, is what I meant by holding on to the front lines, isn't it? So that, I don't know whether colleagues wish to comment on that.
No, I think one of the opportunities for people who are on the curve where they are adopting electronic medical record is to actually leapfrog quite a bit from what people have conventionally struggled with electronic medical records in the formats of how you go about in typing the patient details, and then, of course, tried out OCRs, tried out ambient listening, and all of those other technologies. I think we now are going into a situation where we have a lot of these would be automated by using generative AI. And there are tools that are already available or are at research at pretty advanced stages of research to help in. So putting them into practices would actually catapult a lot of organizations to collect data in an appropriate way and then represent them correctly into any one of the AI systems that they would like to integrate with.
I would just comment that, it really has to be setting specific. If it's a setting that doesn't have EMR, then these tools aren't going to be relevant. But I think the settings that do have the EMR, the idea is that you have a trove of data sitting there, and the point of AI is to try and mine that data in a way that it can take all the complexity of the data and simplify it for human consumption. And that there's an opportunity, perhaps, with this complex data in order to get it simplified to then help the doctor treat the patient.
Excellent. Yes, that's a really good overview, and I would like to hear from the panelists some side in terms of how we see the approach for better integrating the development of AI tools into sepsis, into to ensure that there is long-term relevance and adaptability. What are the challenges that you foresee, and what we expect for the future in terms of what you have promising being presented? And I will start, if I may, by Dr. Carr.
Well, I want to bring in a couple of things here. One is, of course, rightful skilling of individuals who are using AI as a technology, and purely from an implementation science. I mean, you really have to look at the tools that we're going to use, how good they are, how they are relevant, and they go about in looking at the functions that they need to serve, which is the very essence why you would use a tool or give a tool to a physician's hand. But skilling the physicians or the clinicians, nurses, and others to actually utilize this tool are very, very important perspective. So for any other health technology that we have been using in healthcare, we would do an appropriate credentialing, skill the people, then do return demonstration, see how they are doing it, and then go about in giving them a credential to do that. How do we do this in AI? Use of AI is something which is going to be very interesting going forward. There are a lot of people thinking around. And the second point that I want to make is around, the models are aging. There are certain models which have been there for quite some time now. They are aging because of data drift, the shift around the technology itself, and then becoming obsolete. How do you keep your models and use the models which are contemporary and relevant for clinical practice? Those would be the very important things that you need to look at when you adopt AI in your systems.
Thank you, Dr. Carr. Dr. Hosein?
At the back of my mind is business theory and production versus marketing. The production model says, here's a product, please use it. The marketing model says, do I need it? And I guess the emphasis should be on the frontline clinician having information in a manner that they want, that they can use. So whilst we want to upskill people, is that a technological one in handling software? I don't think it should be. It should be, I have a decision to make, as was mentioned by Dr. Shapiro. Should I treat the chest? Is the patient responding with a ventilator-associated pneumonia? Yes or no? Do I need to change? I want information that I can use. And if that information is presented in a manner that is understandable in clinical language, if it's not, then I'm not going to use it because I don't have time to go and, it's a point of decision making now. This is not about an audit after the fact. So I think that as we move more and more into AI, I would like to see how we begin with small things in terms of, I think one of the junior doctors here at my hospital is looking at AI assisting in ward round records and so on. And of course, there's an immediate audit of what was said and what was not said. And the value would be in, well, have we got a proper record or not from the ward round? So I guess in conclusion, my view is that the technology should sit behind elsewhere. It should not be an issue for the frontline clinician who needs the information in a usable manner. The technicalities, I'm not particularly concerned about. Any experienced clinician knows what they want. They know what they just don't have the ability to get it to make a decision. And this is where AI may help in extracting that data, mining that data, pulling it together and saying, well, here is a probability of infection or not. That's my view right now.
Thank you. That's really foresight. Dr. Shapiro?
Yeah, so I think a historic flaw of a lot of these systems is they end up being computer systems built by programmers and in the end of the day for programmers. It's kind of programmers saying, here's what I think the doctors need, so I'm going to program it. And then by the time the doctors see it and have input, it's already a fixed system. I think a strength will be if it's a combined team of clinicians and computer programmers creating systems that can integrate into the workflow of clinicians. So when we get the systems integrated into the workflow, giving the informations to the clinicians that the clinician needs, that's when I think they'll be most effective. So hopefully this will be a way to integrate and to help the clinicians as opposed to fight against the clinicians.
Excellent, excellent. Thank you very much. We have learned how AI can enhance the sepsis detection and the management, and how crucial early detection is in saving lives. And you have given us foresight of what is coming in all settings in terms of what AI will bring into the statistics. We want to thank all our panelists, the audience, for being active in the chat and in the conversation, and also the sponsors of this session for the insightful discussions. We appreciate your active participation, and we encourage you to sign into the World Sepsis Declaration and follow us on social media. Thank you very much for today's sessions. Pleasure.
Thank you very much.